Welcome back to my educational channel on biology. Teacher Janet here. In this video, we will discuss a Form 5 topic which is Development of Pollen Grains and Embryo Sac from Chapter 6, Subtopic 6.2. So here we're going to discuss the formation of the gametes of a plant, a flowering plant. For example, the formation of pollen grains in the anther and also the formation of the egg cell or ovum in the embryo sac, which is a big cell found in the ovule of the flower. Okay, so the ovule is protected by the ovary here. So how is the egg cell formed from the ovule or in the ovule? Let's find out about the formation of the pollen grains and egg cells, which are the male and female gametes respectively in a plant, a flowering plant. The learning standards are as follows. 6.2 Development of pollen grains and embryo sac. Number 1, describe the formation of pollen grains in an anther, and number 2, describe the formation of the embryo sac in an ovule. So this picture shows you the pollen grain with its two nuclei. Okay, and this is the external appearance of different types of pollen grains from different plants, right? So how is the pollen grain formed, and how is the embryo sac that contains the ovum or egg cell formed? Let's find out more. Diagram shows us the overview for Chapter 6, Sexual Reproduction in Flowering Plants. In the first video, we have already discussed the structure of a flower. So the male reproductive structure is the stamen, which consists of the anther and the filament. The female reproductive structure is the carpel, which consists of the stigma, style, ovary, and ovule. Now, in this video, we're going to discuss the development of pollen grains in the anther and the development of the embryo sac in the ovule. For the development of pollen grains in the anther, firstly, the microspore mother cell, which is diploid, undergoes meiosis to form 4 microspore cells, which are haploid. Then, in each microspore cell, the nucleus undergoes mitosis one time and once, and this produces a pollen grain with 2 nuclei, which are the generative nucleus and the tube nucleus. Now, for the development of the embryo sac in the ovule, the megaspore mother cell, which is diploid, undergoes meiosis to form 4 megaspore cells in the ovule. 1 cell develops and 3 cells degenerate. So the 1 megaspore cell will undergo mitosis 3 times. It's the nucleus that undergoes mitosis. So this will form an embryo sac cell, which is a big cell, with 8 nuclei each. All the nuclei are haploid each. So to recap from the previous video, this is a longitudinal section of a flower, and the stamen is the male reproductive structure. It consists of the filament, the stalk, which holds up the anther. The anther is the location or the site that produces the male gametes. Whereas for the pistil or carpel, which is the female reproductive structure, there are 4 parts: the stigma, style, ovary, and ovule. It's the ovule that produces the female gametes, right? So let's discuss further. Firstly, let's discuss the development of pollen grains. 6.2a, development of pollen grains. So this is one pollen grain showing you the tube nucleus, which is haploid, and the generative nucleus, which is haploid, inside the pollen grain. Let's find out how this pollen grain is formed. A, formation of pollen grains in the anther. Now this picture here shows us the anthers in a hibiscus plant. So if you look at one anther, it has 2 lobes or 2 sections. Right? Now these are the 2 lobes or 2 sections. 2 lobes, 1, 2. In each lobe there are 2 pollen sacs. So altogether there are a total of 4 pollen sacs in this anther. Alright, pollen sac is a bag that contains the microspore mother cells, which are diploid. So these cells, the microspore mother cells, will produce the male gametes. Now, in this side here, we can see that when the anther has matured, the pollen sac will burst and release the matured pollen grains on the surface of the anther. Right? So it's usually yellow in color. You can see it like powder on top of the anther. That shows you that the pollen sac has burst. So we will start by discussing what happens to the microspore mother cells, which are diploid in the pollen sac. So in the formation of pollen grains in the anther, we start with the microspore mother cells. Now, the microspore actually refers to the pollen grains, which are tiny, right? That's why it's micro. And then microspore mother cells. So the mother cells will produce the pollen grains. All right, start with the mother cells, which are diploid. Now, microspore mother cells, which are diploid, undergo meiosis. Each microspore mother cell divides into 4 cells, 4 haploid microspore cells. So at first they stick together, so they're called a tetrad, okay, of 4 haploid microspore cells. But later on they will separate from each other. So they form the pollen grains, which are haploid, all right? And then the haploid nucleus in each pollen grain divides by mitosis to form 2 haploid nuclei. The first nucleus is called the tube nucleus, and the second one, which is surrounded by a membrane, is the generative nucleus. It's the generative nucleus that will form, like sort of generate 2 male gametes later on after pollination has occurred. So at maturity, you can see that the pollen grain has 2 nuclei, the tube nucleus and generative nucleus. And if it is pollinated by insects, then its wall will be a bit rough, okay, have a specific pattern to help it be able to attach, okay, to stick to the legs of the insects for pollination. So here's the explanation for what we've discussed just now. Now it can be put in a question form like this. Describe the formation of pollen grains and the male gametes in an anther. 8 marks. Okay, it can be asked as an essay question. So let's go through this very quickly. As the anther develops, a group of tissues grows inside each lobe to form 4 pollen sacs. In each pollen sac, there are hundreds of diploid pollen mother cells called the microspore mother cell. The microspore mother cell divides by meiosis to produce 4 haploid microspore cells, which group together to form a tetrad. Each microspore cell in the tetrad develops to form a pollen grain. Next, in each pollen grain, the haploid nucleus divides by mitosis to form 2 haploid nuclei, namely the generative nucleus and the generative nucleus here and the tube nucleus. When the pollen grain matures, the thick waterproof wall here of the pollen sac breaks, and the pollen grains are released. The application of the pollen grain. The rough surface of the pollen grain helps it to stick easily to pollinating agents, such as insects. Okay, for example, the surface here is rough and may be spiky, so that it's able to stick easily to pollinating agents such as insects and to the stigma of a flower. This is for flowers where the pollinating agents are animals and insects. But for wind pollinated pollen grains, they are usually light, they have smooth surface, and they are small in size so that they are easily carried by the wind to other flowers. Now, after pollination, that means the transfer of pollen grains from the anther of a flower to the stigma of a flower, the generative nucleus divides by mitosis to form 2 male gametes, sometimes they are called male nuclei, in the pollen tube that grows in the style, which is part of the female reproductive structure. Okay, now so we see here, the pollen grains are released when the pollen sac bursts in the anther, and then it may be carried by the pollinator or pollinating agent like the bees, or it can be carried by wind. So in this case, it's carried by the bee to the stigma the same flower or another flower, right? So once the pollen grain lands on the stigma, the pollen tube will grow downwards, right? And here we see the generative nucleus and the tube nucleus. Now the tube nucleus will lead the way down, whereas the generative nucleus will undergo mitosis again to form two male gametes. Earlier there's one process of mitosis in the anther. So now when the pollen grain has reached the stigma, has landed on the stigma, there's another process of mitosis occurring to the generative nucleus. Okay, so generative nucleus undergoes mitosis to form two male gametes, but the tube nucleus does not undergo mitosis in this case. Okay, so these two male gametes in the pollen tube will also travel down with the tube nucleus to the uh, ovule and to the embryo sac in order to fertilize the egg cell or ovum later on. Are some beautiful pollen grains from different plants. We can see that these pollen grains do not have the same shape or size. They differ in shape, size, and also texture. Okay, but basically, if the pollen grain has a rough surface or spiky surface, they are usually insect pollinated. They are transferred from the anther of a flower to the stigma of the same flower or another flower by insects or animals. All right, because the rough surfaces help these pollen grains to easily stick to the bodies of the insects, like the legs of the insects, so that they can be transferred from the anther to the stigma during pollination. All right. So examples of pollen grains that have the spiky surfaces are sunflower, morning glory, and hollyhock. And for lily, it has a rough surface. Okay, the pollen grain has a rough surface. And for the primrose, it has a smooth surface, right? It's three-lobed. It has three lobes. Now, for some of these pollen grains, they may be sticky, all right, to be able to attach to the legs of the insects. Okay, some of them are sticky. Whereas for the last one here, the castor bean pollen grain, you can see that it's quite small in size compared to the other pollen grains. And so it is very light, and then it has a smooth surface. So it can be pollinated by wind and also insects. Okay, when it's smooth, small and light, it's easily transferred by the wind from the anther to the stigma of a flower. And please take note that these are all pollen grains. They are not the seeds. The seeds of the plant is only formed after fertilization. Okay, this is before fertilization. So these are pollen grains. Discuss activity 6.2 on page 118 of your textbook, studying adaptations of pollen grains for pollination. So three types of pollen grains are observed under the microscope, namely hibiscus species pollen grain, Mimosa pudica pollen grain, and lalang pollen grain. So these are the diagrams of the characteristics of the pollen grain, okay, when observed under the microscope. Let's start off with hibiscus species pollen grain. So if you look at it under the microscope, it looks like a mini durian, right? It's round with a lot of spikes on it, just like a durian, a very round durian. So anyway, don't call it mini durian, It's just now how to discuss or describe the pollen grain. It is spiky, okay, it has the hooks here or the spikes, and basically it has a rough surface. So the importance of this characteristic is that so that the pollen grain can easily stick to the insect's legs and surface of the stigma in order to ensure that pollination occurs. Okay. Now, whereas for Mimosa pudica, the surface is not so spiky or rough, but it is sticky, all right, and small, so that it can easily stick to the insect's legs and surface of stigma in order to ensure that pollination occurs. For the lalang, it's different. Its pollen grain is shaped like that and doesn't have a rough surface. Okay, so it is thin, light, and produced in a large quantity. Now this tells us that the method of pollination is by wind. Okay, so that it is easily transferred by wind because of its light characteristic from anther to stigma in order to aid in pollination or ensure that pollination occurs. Now, summary of the formation of pollen grains. First, the microspore mother cell, which is diploid in the pollen sac of the anther, undergoes meiosis to form a tetrad of 4 haploid microspore cells that stick to each other. Okay, so remember, from the microspore mother cell to the microspore cells, right? The microspore here actually refers to the pollen grains. So when the microspore cells separate, they are called the pollen grains. And then the nucleus of each pollen grain, which is haploid, undergoes mitosis once to form the generative nucleus and tube nucleus in the pollen grain. But that's not the end of the story, okay? So because the process is not ended. Then when pollination has occurred and then the pollen grain germinates, that means it grows a long tube on the stigma. Then after the pollen tube starts to grow, there's mitosis again. And that's the mitosis of only one nucleus, which is the generative nucleus, to form 2 male gametes. Each one of the male gametes are haploid. Okay. Now the tube nucleus doesn't undergo mitosis. All right. So the tube nucleus helps to guide the pollen grain pollen tube down to the ovule in the ovary for fertilization with the egg cell. Now let's talk about the development of the embryo sac, right? Which is found in the ovule, in the ovary. This diagram shows you the young ovule inside the ovary of a flower. Okay, so the female reproductive structure of the flower in the flower is called the carpel or pistil. So the carpel is made up of the stigma at the top, the style, which is a long tube-like structure, and then the swollen part here is called the ovary, and then inside the ovary there's the ovule. Let's focus on the ovule. So at the early stage, okay, of formation of the ovule, before the embryo sac or the female gametes are formed, okay, these are the parts that can be seen. So first of all, the ovule is a structure of the flower that's formed inside the carpel, and it develops from a layer of tissues inside the ovary. So a single ovary may contain one or more ovule. Here there's one ovule shown, only one ovule. Now the ovule is attached to the ovary through a stalk called the funicle. Okay, so funicle is a stalk which attaches the ovule here to the ovary wall, right, to the ovary here. And below the funicle there is a placenta. This is the area of attachment of the funicle to the ovary. The function of the placenta is similar to the placenta in humans. It supplies nutrients to the ovule here through the funicle. Then you have the nucellus here. So nucellus is a lump which develops from a mass of tissues inside the ovary. It consists of parenchymal tissue, and the most important thing is that it contains the megaspore mother cell. The megaspore mother cell or embryo sac mother cell is a cell in the nucellus which will develop to form the mature embryo sac with the gamete, the female gametes. Okay, female gamete. So the embryo sac is a big cell that's formed, and then inside is the female gamete. All this develops from the megaspore mother cell. Okay, then we have the 2 integuments on the outer part of the ovule. This is to protect the ovule, and later the integuments will form the seed coat when fertilization has occurred. Okay, so basically integuments are to protect the ovule. And then at the ends of the integuments and somewhere in the center here, there is a small opening called the micropyle. So this micropyle functions to allow the entry of air and water into the seed that will be formed here. All right. the ovule will finally become a seed after fertilization. So when the seed needs to germinate and grow the shoots and roots, then water and air is needed, right? So you enter through the micropyle, okay, a tiny opening at the end of the integuments. So now we're going to focus on the megaspore mother cell, which is diploid, that is found in the nucellus here. It's also called the embryo sac mother cell. This is the cell that will produce the ovum or egg cell or female gamete. So 6.2b, development of the embryo sac in the ovule. These are the notes, and we have discussed this in the previous slide, okay? So you can read through this on your own or copy down the notes. Now this diagram here shows us the functions of the various parts related to the ovule. So you can copy this down and to be able to answer questions on the functions of parts like the micropyle or integuments or placenta. It's similar to what we have discussed in the previous slide. Let's go on to the most important part here, which is the development of the embryo sac in the ovule. The embryo sac is a big cell that contains the ovum or female gamete or egg cell. Okay, so start off with the megaspore mother cell that's found in the ovule. So this megaspore mother cell or embryo sac mother cell is a diploid cell compared to the microspore mother cell. Microspore mother cell gives rise to the pollen grain, which is a tiny cell, whereas the megaspore mother cell will produce the embryo sac, which is a big cell. That's why it's called mega, big, megaspore mother cell. Okay, so the mother cell will produce the gamete, right? That's why it's called mother cell. Now, the megaspore mother cell undergoes meiosis, first of all, to produce 4 haploid megaspore cells. Okay, so megaspore cells are produced from the megaspore mother cell. But 3 of these megaspore cells degenerate, right? And only 1 will then develop. Unlike the formation of pollen grain, where all the microspore cells develop. So here, 3 cells degenerate and 1 megaspore cell develops. So the nucleus of the cell, okay, first of all, the cell will grow, okay, and the cell that grows is called the embryo sac, but it's not matured yet, okay, embryo sac or megaspore cell, which is haploid. Then the nucleus of this cell will divide by mitosis 3 times to produce a large cell with 8 nuclei. Let's look at the next slide. So let's continue with the discussion on the embryo sac, all right? So after the formation of the megaspore cell, the nucleus of each megaspore cell will divide by mitosis 3 times to produce an embryo sac with 8 nuclei, as seen at the end here. Now, each time the nucleus divides, it will divide into 2, okay? So, from one nucleus, after the first process of mitosis, 2 nuclei are formed. Then, again, through mitosis, 4 nuclei are formed, okay? 2 times 2 equals to 4. And lastly, by the third process of mitosis, 4 nuclei become 8 nuclei. 4 times 2 equals 8, right? Then these 8 nuclei are arranged in a special order or format or arrangement. 3 nuclei move to the top here, and they are called the antipodal cells. They are surrounded by a membrane each. Then 3 other nuclei move to the bottom of the big cell here, and they are called, 2 of them are called the synergid cells, and 1 is the so-called princess or egg cell, all right? It's the female gamete that will take part in the fertilization process. Then 2 more nuclei will be in the center of the cell. Okay, they are called the 2 polar nuclei. So all these nuclei are haploid, right? Now we do not need to know the function of the antipodal cells or the synergid cells, but we must know that the egg cell or ovum and the 2 polar nuclei are involved in fertilization, a fertilization process called the double fertilization. for flowering plants. And this big cell here is called an embryo sac. It's a matured embryo sac with 8 nuclei. So there's no embryo formed yet, because no fertilization has occurred. But in the future, it may form the embryo. Okay, it may have an embryo inside. So it's called the embryo sac. Okay, even before fertilization. So remember, the embryo sac is a big cell with some cells inside, including the egg cell, and it also has 2 polar nuclei. So this diagram here shows us the embryo sac and its position in the ovule. All right. Now here are the 3 antipodal cells above, the 2 polar nuclei in the center of the embryo sac, and then below here we have the 2 synergic cells with the egg cell in between. So normally the egg cell is quite near to the micropyle because the pollen tube will grow and penetrate the embryo sac. Okay, once it grows through the micropyle here. All right. Then the male gamete will come out and then fertilize the egg cell here. All right. So the other parts have already been discussed. Now these are the notes for the discussion on development of embryo sac in ovule. Describe development of embryo sac in ovule, 8 marks, right? So let's read through very quickly. The megaspore mother cell, which is diploid, divides by meiosis to produce 4 haploid megaspore cells. 3 of the megaspore cells degenerate, and only 1 megaspore cell, which is haploid, develops. The nucleus of the cell then divides mitotically 3 times, not just once, 3 times, to produce a large cell with 8 nuclei. So this large cell is called the embryo sac. Now 3 nuclei move to one end of the embryo sac to form 3 antipodal cells. Another 3 nuclei move to the opposite end of the embryo sac to form the 2 synergic cells and the egg cell. Okay, they are covered by an extra membrane. And then each covered by an extra membrane each, by a membrane each. Then we have 2 nuclei in the center of the embryo sac, which will form the 2 polar nuclei. Okay, so the 2 polar nuclei and the 1 egg cell here are all involved in the double fertilization process of the plant, which we will discuss in the next video. So here's a quick summary of the formation of the embryo sac in the ovule plus the ovum or egg cell. Now you can copy this down on a piece of paper and stick it on the wall or the back of your door and constantly review to be able to memorize the terms here, okay, and the processes here. Now the megaspore mother cell or embryo sac mother cell in the ovule, which is diploid, undergoes meiosis once to form the megaspore cell or embryo sac with 1 haploid nucleus. So 3 megaspore cells will degenerate and only 1 megaspore cell will develop, okay, for each megaspore mother cell that undergoes meiosis. So after meiosis, the megaspore cell will grow and then it carries out mitosis 3 times to produce the mature embryo sac, which is a big cell with 8 haploid nuclei, and the arrangement of these nuclei are 3 at the top, 3 nuclei at the top, 2 in the center, and 3 nuclei at the lower end, including 1 ovum or egg cell, which is haploid, and 2 polar nuclei, which are haploid each. So these 3 nuclei will be involved in the double fertilization process later on with the male gametes. So we've already discussed the formation of the pollen grain, which will later contain the male gametes, and also the formation of the embryo sac, which contains the female gamete. Now let's continue with a comparison of insect or animal pollinated flowers, such as this flower that's very brightly colored, and the wind pollinated flower, such as the flower of lalang, right, which is a type of weed or grass. So this is also discussed in your textbook, so it's quite important. Important comparison of insect or animal pollinated flower and wind pollinated flower, right? So firstly, for insect pollinated flower, The pollen grain is sticky, rough, or spiky so that it can easily attach itself to the bodies of insects. Whereas for grass or wind-pollinated flowers, the pollen grain is small, smooth, and light so that it is easily carried by the wind from an anther to a stigma. Next, number 2, a moderate amount of pollen is produced for insect-pollinated flower, but a large amount of pollen is produced for wind-pollinated flowers. Okay, because the wind will blow and scatter the pollen grains to many parts, okay, to many places, so it may be lost and may not be able to successfully land on the stigma of the same species of flower. So large amount of pollen grain is produced for wind-pollinated flowers. Number 3, for insect-pollinated flowers, they have large and brightly colored petals to attract the insects. Whereas for wind-pollinated flowers, the petals are usually small, dull colored like brown or greenish color, green in color. Number 4, animal-pollinated flowers have sweet scent and nectar, as you can see here in the nectary, which is the place where the nectar or sweet fluid is produced, right? So the insects will be attracted to the flowers and they will burrow deep to get the nectar, to drink the nectar from the nectary, right? So this is the way that the flower attracts the insects so that its pollen can be transferred to the insects' bodies. Now, for wind-pollinated flowers, they do not depend on insects, so they don't have any sweet scent and any nectar to attract insects. Number 5, for animal-pollinated flowers, the anthers are inside the flower within the petals, all right, and they are firmly held. Same with the stigma. They are inside the flower and they are sticky. So for insect-pollinated flowers, this helps the flower to have its pollen grains transferred to the body of the insects, especially when insects burrow deep to get the nectar. Okay, the bodies will brush against the anthers, and the pollen grains will then be easily attached or stuck to the flowers, to the bodies of the insects, and also the pollen grains can be transferred easily to the stigma, which has a sticky surface. Okay. Whereas for wind-pollinated flowers, the anthers are outside the flower, loosely held, so that they are exposed to the wind. Then the pollen grains can be easily blown to other places. The stigma is also outside the flower and it's usually feathery, like a net, in order to catch or trap the wind-pollinated pollen grains. Okay, so examples of insect-pollinated flowers are the rambutan, durian, papaya, rose, sunflower, and hibiscus flowers. For wind-pollinated flowers, examples are corn, grass, and paddy. So here's a table showing the comparison of insect-pollinated and wind-pollinated flowers. What are their adaptations? Okay, what are the different adaptations? So it's the same as what we've discussed before, but here I put in the reasons why. Okay, so we'll read through very quickly. Pollen grain structure for insect-pollinated flower. The pollen grain is sticky or has a spiky surface or rough surface in order to stick to the bodies of insects well. Okay. Now, for wind-pollinated flower, the pollen grains are small, smooth, and light so that the pollen is easily carried by the wind to other places and to the stigmas of flowers, and it does not clump together because it's not sticky. Next, the amount of pollen produced by insect-pollinated flower is a moderate amount because insects can transfer the pollen grains more effectively than wind does. Now, for wind-pollinated flower, a large amount of pollen is produced because most of the pollen may not reach the another flower of the same species, right? Now, petals. Petals for insect-pollinated flowers, they are large and brightly colored. So insect-pollinated flowers have large and brightly colored petals to attract insects such as bees and butterflies. But for wind-pollinated flowers, the petals are small, often dull green or brown, because there's no need to attract insects. So insect-pollinated flowers have sweet scent and nectar to attract insects. But for wind-pollinated flowers, there's no scent or nectar, because they do not need to attract insects. Anthers in the insect-pollinated flowers. Okay, they are found deep inside. They are found inside the flower and held firm in position to allow the insects to touch the anthers and brush against the anthers when they search for nectar. And thus, this can deposit the pollen grains on the insect's body. But for wind-pollinated flowers, the anthers, as you can see here, are loose and held on long filaments so that they can be exposed to the wind and release the pollen grains into the wind easily. Stigma for insect-pollinated flowers are inside the flower and they are sticky to allow insects to touch the stigmas when looking for nectar and to deposit pollen on them, on the stigma. Okay. Now outside, for wind-pollinated flowers, the stigma is held usually outside the feather, outside the flower. It's feathery or net-like to catch the pollen that is drifting in the wind. So let's test ourselves. Now, question 1. Some of these questions are related to the questions in your textbook. Now, firstly, the picture shows an insect-pollinated flower. Explain how the position of the anthers and the stigmas enable pollination to occur. Okay, 3 marks. Pause the video and try to answer the question yourself. Explain how position of anthers and stigmas enable pollination to occur. So firstly, we must explain the position of the anthers and stigmas first. We must take the position first and then explain how it enables pollination. Okay? So the anthers and stigmas are located inside within the petals of the flower and they are firmly held. This allows insects that enter the flower to search for nectar to touch the anther and stigma easily. Thus, the insects will come to contact with the pollen grains from the anther, and after that, they will transfer them to the stigma of the same flower or another flower. Question 2. The development of pollen grains involves both meiosis and mitosis. So one process of meiosis and then one process of mitosis. Now explain the importance of both processes to yourselves. Why is it that in the formation of pollen grains there is meiosis and mitosis of the mother cells, the diploid mother cells? Now answer. Firstly, the process of meiosis functions to produce haploid gametes from the diploid parent cells. So in meiosis, the number of chromosomes will be become half, right, compared to the diploid parent cells. So the gametes produced will be haploid instead, having half the number of chromosomes as the parent cells. And parent cells are actually the microspore mother cells. Now, how about the process of mitosis? It produces 2 haploid nuclei. Okay, from one haploid nucleus, the process of mitosis occurs to this one haploid nucleus to produce 2 haploid nuclei, which are the tube nucleus and the generative nucleus. Both of these are important for the double fertilization process, right, which occurs in the female reproductive structure after pollination. Now, the tube nucleus guides the growth of the pollen tube to the embryo sac in the ovule. That we will discuss later on in another video. Whereas the generative nucleus further divides by mitosis to form 2 male gametes. That's after pollination. And one of the male gametes will then fertilize the egg cell to form the zygote. Again, we'll discuss this later on. Now, in this last section, we are going to discuss the Gymnosperms, which is another type of plant that is separate from the Angiosperms of flowering plants. Okay, so this is discussed a little bit in your textbook, so we need to know a little bit about them. So basically, Kingdom Plantae consists of all types of multicellular plants, which contain chlorophyll, and they can synthesize their own food by photosynthesis. Okay, So these are plants that are quite usually quite well developed, and they can undergo asexual or sexual reproduction, right? So examples are the seedless plants like the ferns here. These are the ferns, pakupakis, in Malay we call it pakupakis, and also the mosses. Mosses are tiny plants that grow in the moist regions and also on tree trunks and rocks, and also on soil. So these are all seedless plants. Now, plants that produce seeds, there are two categories, the angiosperms and the gymnosperms. Okay, so angiosperms are all these flowering plants that produce seeds and fruits also, like papaya. Okay, and then we have the gymnosperms as shown here. All right, these are different because they don't produce flowers, and their seeds are not protected by the ovary or not protected in the fruit, you know. So let's look at the difference between angiosperms and gymnosperms. Now here I have given you an overview of the plant kingdom or kingdom plantae, which consists of tiny plants called the mosses and liverworts. These are not so advanced. Ferns, so all these are seedless. They don't produce seeds. Ferns produce spores. Now, then we have the angiosperms and the gymnosperms. They produce seeds. Okay, so angiosperms are flowering plants, and we are studying about the fertilization process in angiosperms. Okay, the reproductive process in angiosperms. But besides that, there's another category of plants called the gymnosperms. Gymnosperms, for example, the conifers. For example, the tree that's used to celebrate Christmas. Okay, the Christmas tree, right? That is a gymnosperm. The shape is more or less like a triangle. Okay, so this includes the pine tree and the fir tree, right? Usually the shape is like a bit triangular with a sharp peak at the top. And they don't produce flowers, they produce cones. Okay, let's find out more about gymnosperms. Now, the difference between gymnosperms and angiosperms, you have to know a little bit about it. So angiosperms are flowering plants, whereas gymnosperms are non-flowering plants. Angiosperms have reproductive structures called the flowers, which produce the gametes, the male and female gametes. Gymnosperms have reproductive structures called the cones, which produce the male and female gametes, right? They don't have flowers, they have cones, as shown here, cones. Now, angiosperms. For angiosperms, the pollen is transferred or pollinated by animals, wind, and water, but for gymnosperms, the pollen is transferred or is pollinated by wind, okay, by wind usually. For angiosperms, the seeds are enclosed in the ovary or fruit, but for gymnosperms, the seeds are not enclosed in the ovary or fruit. It is naked. We call it the seeds, naked seeds. They're exposed. They don't have a covering like the fruit, okay, to protect them. Then angiosperms, they have flat leaves, such as those shown here. Okay, they have many shapes, whereas gymnosperms have needle-like leaves. Okay, pointed leaves like that. So examples of angiosperms are rose, hibiscus, grass, and for gymnosperms, the pine tree and fir tree, okay, as shown here. So plants of the gymnosperm category, such as the pines and the fir tree, do not produce flowers. However, these plants produce pollen and egg cells through the structures called the cones. So there are two types of cones, and they have different shape. The pollen cone, as shown here, is usually longish. It's the male reproductive structure. It has scale-like structures that produce the pollen grains. Okay, so if we cut through the pollen cone or the male cone, we can see that they have scales. Inside the scales, the male gametes are being produced, all right, which are actually also called pollen grains, right? And then, the pollen grains contain the male gametes. Then we have the female cone, which is sometimes called the seed cone. This is the female reproductive structure. It can be on the same tree, right? And usually it's higher up. So it has scale-like structures that produce ovules and egg cells. Again, these are the scale-like structures. So you cut through the seed cone or the female cone. Now you can see here, the ovule is found underneath the scale here. See, all these are ovules, waiting to be fertilized by the male gamete. Okay, the egg cells are in the ovule, and the egg cells are waiting to be fertilized by the male gamete from the pollen cone, right? So there's no fruit to cover uh, the seed with its spawn. Okay. Now the seed cone is larger than the pollen cone. Okay, seed cone is a female cone, is larger than the male cone or pollen cone. Normally, both pollen cones and seed cones are produced on the same tree, but the seed cone is usually higher up, so as to avoid self-pollination, or avoid the pollen from the same plant fertilizing the male gamete from the same plant fertilizing the uh, female gamete from the same plant, is to avoid that. Okay, so the seed cone is usually higher up than the pollen cone. This shows the life cycle of a gymnosperm tree. So the pollen cone or male cone produces the pollen grains, which contain the male gametes, and the pollen grains are transferred by wind, okay, in the process of pollination to the seed cone or the female cone. So when the male gametes fertilize the female gametes in the ovule here, in the scales, then the embryo is formed after fertilization. The embryo is found inside the seed, okay, it's the young plant. So if the seed falls on the ground and it grows, then it can form the next generation, which is a new tree, an adult tree. Okay, it can grow into an adult tree. Now this picture shows you the seed cone and which is the female cone, and the pollen cone, which is the male cone, right? And this picture here shows you for each scale in the seed cone. Here is an ovule of in the scale. Now the pollen grain, if it lands on the scale, will grow a tube or so, and then the male gamete will fertilize the uh, female gamete in the ovule. Okay, and then after that the seeds are formed and they are dispersed, usually by wind, and then they grow into mature trees. Here is some further information, advanced information that some of you may be interested in. So here we see the seed cone or the female cone with the ovules already formed in the scales. Now, after fertilization, when the male gamete has fertilized the ovum inside the ovules, the seed cone will grow bigger, and then the seeds are produced through fertilization. So the seeds will be resting on the scales like this, right? So if you look at one scale, you can see that for the pine tree, it has 2 winged seeds on one scale. Okay, these are the seeds. And the seeds have wing-like structures. So what are these for? All right, so you can see the wing-like structures here. Okay, these are the seeds that have been separated out. Okay, this is the seed with the embryo or the young plant inside. The seed has not germinated, right? And then you can see the wing structure here. So firstly, the seeds of the pine tree, they are not enclosed in an ovary or fruit. Okay, there's no ovary or fruit wall to protect the seed. So it's called the naked seed. That's what its name, Gymnosperms, implies, right? So these naked seeds have wings, wing-like structures that aid in the dispersal by wind because of these shape structures, these wing-shaped structures. There's larger surface area, and this will help the seed to float in the wind, all right, so that it can be dispersed or scattered to a place further from the parent plant. Thanks for viewing, and I hope that you have benefited from this lesson. In the next video, we'll discuss the important process of double fertilization, which is a key factor in the success of the Angiosperms on the earth today. Please do share, like, comment, and subscribe to my channel. And all the best to you. Till we meet again, goodbye.